Great. I'd like to welcome everybody, both here in Wilmington, North Carolina, and at home participating through the webinar to the SoulCap workshop. When Dave Douches asked me to speak about breeding in the genomics era at the Potato Association of America meetings, I kind of scratched my head. I guess the conclusion I came to is that Dave wanted me to convince you all that potatoes always taste better with ketchup. Seriously though, there are aspects of the crops, potato and tomato, that create synergy. So when you heard Robin talk about the genome sequence and the SNP discovery pipeline, that's a place where those of us in the tomato community have really benefited from her expertise and the pipelines that were developed for the potato part of this project. On the other hand, when we start to look forward and say, where are we going to go with this technology, I think there's a place where the tomato community has something to offer to the potato community, in that marker-assisted selection has been used in tomato breeding since the 1970s, where isozyme linkage to disease resistance was routinely used. And so we've been thinking about marker-assisted selection for a long time. Now, I will acknowledge that there are major differences in the crop. We have a diploid inbreeding species that's usually commercialized in the form of hybrids versus a highly heterozygous tetraploid that can be clonally propagated. Those represent major differences, but I think some of the overarching concepts that I'm going to talk about are applicable. And so what I really want to do in this talk is to be somewhat provocative, just to get you thinking about the future and how, as plant breeders, we should be thinking about modifying our programs to make use of the accessibility of the genomics data. So having given that brief introduction, I also want to point out that I will be talking some about the Illumina data that we've had access to, both from the next generation sequencing and the array. When I talk about the array, I really have to acknowledge Mathilde Kaus from INRA in France and Martin Gunnell from Trait Genetics, as they also help to add content to the consortium chip. I also want to take a little bit of time to acknowledge Jeanette Martins from the Seed Biotechnology Center at UC Davis, who's helped put this workshop together, as well as John McQueen at Oregon State University. So John is the person that's making sure that this broadcast gets out to our audience that's participating online. Okay, just as an overview, what I'm going to be talking about is a little bit about breeding strategies with a focus on the kinds of populations and emphasizing the size and the structure of those populations. And I will touch a little bit on multiple trait indices. I'll talk about the genotyping strategies and some of the tools, but the main focus that I really want to spend most of the time on is this area of integrating the breeding strategies and the tools. Okay. This slide sort of tips my hands a little bit to indicate what I think are some really important concepts and equations for plant breeders. And what I'm trying to emphasize with this slide is that as plant breeders, we work with populations that are often grown under multiple environments. And so I'm showing you some slides of some of our breeding trials in Ohio, and you can see that some of them are under very dry conditions and some of them are virtually underwater. And I think I should point out that in some cases we can get this all in 1 year. But as breeders, we're interested in that interaction between genotype and environment. And so when we think about our science, it's heavily based on the ability to separate variants into components, environmental components and genetic components. And really, so heritability is all about that. The gain under selection equation relies on separating variation into genetic and environmental components. When we talk about relative efficiency of selection, again, we're really talking about heritability based on trait versus the indirect selection, which would be molecular markers and their genetic correlation with a trait. And then finally, it's also important to realize that when we think about the significance of a marker associated with a trait, that the F-test really boils down to this last equation where N is the population size, R is recombination frequency, and then G is an estimate of the proportion of genetic variation explained by that QTL. Okay, so just to kind of rehash this, we need to be able to partition variance into phenotype and genotype. When we think about gain under selection, indirect selection is rarely going to be more efficient than direct phenotype selection. The only time where we're really going to see marker-assisted selection leading to greater gain is when we can accelerate the breeding cycle. And I think this is something very important for the potato community to think about. I think in the tomato community we've already kind of embraced this. But the main point is that it's very rare for a marker to explain more variation, more genetic variation than the direct phenotypic measures. Okay, and so if you're talking about gain under selection, the only way that marker-assisted selection is going to be more efficient is if you can get a couple of generations per year and accelerate the process, right? And then again, when we talk about what the statistical test for a marker association with a QTL is, it's dependent on the recombination distance between the marker and the QTL, or the gene, if we want to get it down to that level, and the strength of that QTL, in other words, the proportion of genetic variance that QTL explains, and then finally the population size. Okay, so we can't control the proportion of variance explained by the QTL, but we can control our population sizes, and to a certain extent we can now control the recombination distance because we have such an abundance of markers to work with now that it's not that hard to come up with markers that are closer to the trait. Okay, so these are themes that will echo through the rest of the talk. So the take-home messages that I want to leave you with, and I'm going to give those towards the beginning, which then allows you to either check out or go to sleep, or maybe stimulate your interest in listening to the rest. So as plant breeders, we need to really keep our breeding programs focused on population development and phenotypic evaluation. The limitations to integrating genotyping and breeding are population sizes, specifically how population sizes affect recombination, but also in terms of the power we get to test for associations. And then the other limitation is our ability to phenotype large populations and to do that well. I think one of the themes that Dave emphasized in his talk, and I will kind of re-emphasize, is that we're getting to the point where the marker work can be very efficiently outsourced. Right? We don't all need to run a genotyping center out of our lab. Okay? And this really has profound implications on how we train our students. For those of you that have been using markers in your program, you probably bring in students and you have that student work for a couple of years collecting marker data, and maybe if they're lucky they've got a couple hundred markers in a 2-year period, and then they move on to the analysis phase, et cetera, et cetera. Well, Dave just talked about how you can, in theory, come up with 8,000 markers on a population in about 3 days, right? So our students aren't going to be spending large amounts of time getting the marker data. What we really need to have them doing is thinking about population structures, phenotyping well, and then analyzing data. Okay? And then finally, I think most of you have probably heard about association mapping. The next phase of this becomes genomic selection or genome-wide selection. And the success of genome-wide selection is based on robust models of phenotype that include multi-trait selection indices. And I think as breeders, rather than worrying too much about the statistics behind genome-wide selection right now, what we really need to think about is our selection models. How much do we weigh the different traits? And I think the best example I can give you for that, and I'm sure there are parallels in potato, is that most people will tell you that the most important quality trait in a processing tomato is soluble solids. But I would challenge you to find somebody that actually pays for that. And if people aren't paying for it, then how can we weigh it in selection? Right? And so we really have to get the industry goals and desires in sync with economic models and in sync with our selection strategies. Okay. So the question came, do they not pay for it, or do they only take varieties that have a high soluble solids anyway? I think if you look at trends over time, it would be hard to make a case that we have improved soluble solids in tomatoes. Okay. So, all right. So what I want to emphasize again is that for the plant breeders, what we really need to do is be concerned about phenotype. Okay. So what I want to do now is I want to shift gears and talk a little bit more about populations and population structure. Typically when we think about populations in the context of academic research, we're thinking about biparental populations. So in tomato that would be F2s or rills or back crosses. In potato you're doing most of your mapping in F1s. Okay, so two heterozygous parents are crossed together and then you've got your mapping population. But in the context of a breeding program, most programs have population structures that consist of many independent crosses. Okay, and I think that the lesson that we can learn from association mapping is that we do in fact have tools where we can start thinking about linking markers to traits, not in the context of a single biparental population, but in a more complex population. Okay, and I'm just going to run through some of the standard populations and then some of the populations that are now being used for what can loosely be called association mapping. So again, you know, for inbred crops, F2s are standards. For outbreeding, you know, crossing two parents together and looking at the segregation in the first generation. Rills and backcross progeny, advanced backcross and inbred backcross progeny have all been used. I think we could call those standard at this point. Emerging are natural populations, unstructured populations, family-based populations, chain crosses, and then nested association mapping populations, which are essentially nested rill type populations, right? And I think some of these structures or ideas could easily be adapted to potato breeding populations. Okay. So, you know, some of the populations that have been standard have the advantage of having a strong theoretical basis. In other words, there is off-the-shelf software that can handle the population structures inherited in them. They have balanced designs, so you don't have skewed numbers in the different marker classes. There are tools for interval mapping, so you know, really what interval mapping brings to the table is it changes the point of analysis from a single marker to a pair of markers and then incorporates recombination distance. The problem from a breeding perspective is that most breeding programs don't collect extensive data in those early generation populations. So in a tomato breeding population, it would be extremely rare to collect a lot of data on individual plants in that first generation. And then the other thing is that these standard populations often retain way too much donor parent. Okay? And this is particularly a problem with integration. And I know that there's at least a couple of people at this meeting that are doing integration work to bring in new resistance into potato. And so in inbred species where we're doing seed transmission, it's very common now to use advanced backcross or inbred backcross type populations. And these have some advantages in terms of reducing donor parent and creating a nice uniform genetic background, but then they have disadvantages in terms of an unbalanced design relative to the marker classes, and therefore also very limited power to detect epistasis. And so those limitations have really fueled the interest in the unstructured populations, because these are really populations that are more like what we would use as breeders. They may be unbalanced and therefore have some of the disadvantages of an unbalanced population, and we need to correct for structure, and I'll explain that in a little bit. Okay, so what I'm trying to do here is just give you an overview of some of the pros and cons of some different population approaches, just to get you thinking about that. Okay. I'm now going to switch gears into talking a little bit about how we integrate phenotyping. So one of the messages that I want to leave you with is that, in my opinion, we need to be more objective and quantitative in our phenotyping. I definitely think that's true for the tomato community, but I also believe that it's true for the potato community. And as part of that, because we're also talking about creating databases that can be used over a longer period of time, we need to start thinking about linking our phenotypes to ontologies. In other words, creating phenotypic categories that facilitate storage in databases. The genotyping I'll cover a little bit in terms of SNPs and sequencing. This is an area where technology is changing. So, we've talked about the Infinium arrays that have been developed, but it's getting to the point where genotyping by sequencing is almost going to be more cost effective. Okay. And so, again, what I want to emphasize is, as breeders, what we need to be thinking about is the population size and experimental designs that we use. So then the next step is linking phenotype to sequence variation, and then finally using this information for selection. Now, the reason that I've put 3 of these categories in green is because, as breeders, if we concentrate on those areas, we know we can make progress. Okay? And so some of the influence of genomics will help us to make traditional breeding more efficient, even if we ignore the genotyping and the map-based work. Okay, so that's a message that I want to leave you with. Okay, so what I'm going to do now is talk a little bit about evaluation and phenotyping, and I'll talk about some specific examples in tomato, but it's the general concept that I want to leave you with about trying to make our phenotyping more objective and more quantitative. Okay, so what I want to point out is that genomics has introduced this concept of storing data for communities, right? And it's now gotten to the point in human genetics and genomics where there is an emphasis on also storing phenotypic data. That trend is moving rapidly into plant sciences and plant breeding. It's going to get to the point at some time in the near future where if you want to publish a mapping study, as an example, you are going to have to make your phenotypic data available in some sort of a database format. Okay? It's important for us to recognize that. But more importantly, from a breeding perspective, having archived phenotypic data is going to give us more power to work across years and environments. Okay. So if we're going to be storing data, it requires that we agree upon criteria and scales. And what I've done in this slide is kind of take a historical perspective and look at different types of objective phenotyping. So if we look at the system that the National Plant Germplasm System uses in GRIN, it's a 1 to 9 classification system for all data. So for traits such as color, we would assign a numerical variable that matches a color. So 1 is green and 2 is yellow. All right. That's okay to a certain extent, but there's some flaws with it if you think about quantitative analysis. Now, That descriptor system is also used for attributes for fruit shape, just like it is for tuber shape and size, right? And so, you know, a 1 might be assigned to flat, a 2 to round, 3 to ellipsoid, etc. The problem with these systems is that there's not necessarily any kind of a linear relationship, right? So 1 plus 2 does not equal 3, okay, which then affects the kind of statistics that we can do on these data. So more recently, there have been systems that have been developed that are objective but also quantitative, so that the measurement can be interconverted and data can be obtained very efficiently. I'm going to give you some examples of these. So the other important thing about having really truly objective data, not scaled data, is that the estimates of variation that are obtained are also very meaningful in terms of estimates of heritability. Okay, so in the case of tomato fruit shape and fruit color, Esther Vandernap's group has developed the tomato analyzer software, which works off of scanned images and then can collect highly objective data that can be used to look at attributes of size, shape, and color. And it turns out that these attributes can then be overlaid very nicely with the UPOP descriptors. You can quantitatively show that, okay, of the 8 original classes, 7 of them are quite robust statistically. All right. My group has spent a lot more time looking at color because that is a trait that has an economic value. And so instead of using a 1 is green, 2 is yellow type of system, we've worked off of quantitative systems. We've tended to work more with the universal C-Lab LAB system, but we've done work to convert the computer-based RGB into that in a way that gives us very good linear correlations. So we can collect data from different parts of the tomato fruit. We get good linear correlations between different color systems, and it gives us very, very objective measurements that we can then use for selection purposes. Okay. Now, furthermore, this data can then be organized in a way that facilitates retrieval as well as placement into databases. Okay, and so what I'm showing with this screenshot is the ontology browser at the Solanaceae Genomics Network, or SGN. And you can see the Solanaceae phenotype ontology in about the middle. I'll get my mouse right there. Okay, and if you click on that, you can see some of the different ontologies that are there. You can click on fruit, fruit color, and then different aspects of where the color is measured, and finally you can scroll down to, say, average hue as a trait. Okay, so the traits we collect are database. Now, this is what it actually looks like when we collect it into a template, right? So it's a flat file. It's linked to an image. There's some information on experimental design, and then all of the traits that we're collecting are linked back into the ontology for databasing purposes, right? And so units are there. All the information we really need is there. Okay, and then again, one of the principal questions we're interested in asking is, with this kind of data, is there in fact a genetic signal? Right? Are we working with a heritable trait? Right? And if the answer to that is yes, then we have something that we can make breeding progress on, whether we're working with markers or not. Okay. All right, and I think that the Eastern United States evaluation trials are a good example of a nice start with a database that's going to serve the potato community well. And what I specifically want to mention about a database like this is that you're going to build up a multi-year data set. And this multi-year data set is not always going to have the same set of genotypes. It will have overlapping genotypes. But by using the power of, say, BLUPs, you will be able to do marker trait analysis on these multi-year data sets. And so if the varieties that are in this trial are genotyped on a yearly basis, only the new ones, of course, because you don't have to keep re-genotyping, you're going to build up a data set that then serves you well as essentially a training data set to look at marker trait associations and models for genome-wide selection. Okay? But it's very important to think hard about the data that's going into the system, making sure that it's standardized across locations and as quantitative as possible. And really your model for this is to look at what the dairy industry does, right? Where yield is measured in gallons per day per cow, and that can all be linked back through pedigrees to specific dams and bulls, and you can predict the performance of a bull. You can say that this bull is likely to produce progeny with this sort of a milk yield, right? So that's the beauty of this kind of a system. Okay. I'm going to just shift gears now and talk a little bit about genotyping technology, but really the message that I want to leave you with is that genotyping technology is changing rapidly, and that markers really aren't the limitation anymore. Okay, what this slide is illustrating is the scheme for creating next generation sequencing. So you create a library, that library goes through some quality control, it then gets sequenced in a flow cell. That sequence data is basically image intense, and that image data is then deconvoluted. You go through an assembly process, and you've got data that can then be mined for polymorphisms. Okay, so in the context of SolCap, we mine that data for polymorphisms for the Illumina array, but sometime in the very near future, you're going to do your SNP calling based on this kind of data. Or more importantly, somebody's going to do it for you, right? Okay. You know, what I'm going to do here is just go through really some of the statistics on the tomato chip, and I want you to see how well they parallel the potato chip. So again, we were working with a 10,000 probe array. We had 8,784 SNPs. So some of those SNPs had to use 2 probes to score. 7,627 passed quality control. We cross-validated with the INRA and the trait genomics data set. There were community candidate genes, and that many of these SNPs work within cultivated populations. It also turns out that they work very well with wild populations, and I know that that was a question, but over 3,700 of these SNPs have been mapped in a reference population that's cultivated by wild species. Okay, so it's like in potato, the tool is working both for cultivated as well as wild species. I guess the other thing that I want to leave you with is that, again, because of the pipelines that Robin's group developed, and because of some other tricks that we use in terms of using allele frequencies in making decisions about our content, we had a very, very high validation rate. You know, essentially overall the validation rate was over 97%. Okay, so it's a very robust set of tools. And then if we start to look at pairwise comparisons, there are simply a lot of SNPs available even for biparental crosses, and that's really what the theme of this slide is. So you can look at the pairwise comparisons and see that there's a lot of markers to choose from. So these Illumina arrays are excellent survey tools. And in terms of how one would use a tool like this, I think what we need to do is give a lot of thought to the germplasm that we're going to genotype. Is that going to represent a regional breeding program, like all of the Northeast for potatoes, or is it going to represent your personal breeding program? Right? We have to think about what comparisons we want to make, which is influenced by the questions we want to ask of the data. So in the case of potato, you might want to ask, I know that many of the potato breeders run programs that create material for both fresh market and processing, right? So you might want to ask whether you actually have differences in those germplasm classes. Do you have structure that correlates with those different aspects of your program? And then finally, really the goal is to ask what sequence variation is important to the phenotypic variation that we see. Okay. Another bit of information that can come out of these survey tools is that we can begin to look at the distribution of variation along chromosomes. And what this slide is showing is just 2 chromosomes for tomato, chromosome 1 and chromosome 2. There are 3 germplasm classes illustrated here. Vintage material, which is what we might call heirlooms and land races, fresh market material, and processing material. And what you can see is that there is differentiation between the germplasm classes. So there are portions of chromosome 2 that remain polymorphic in processing tomato, but have been completely fixed in the fresh market germplasm that we looked at. Okay, these are the kinds of features that you're going to see that represent structure that are inherent in either market class breeding or even regional adaptation. Okay, now another thing that you're going to see is population structure or subdivision that is based on some aspect of selection. Okay, and so when we run a Bayesian analysis using the program called structure, what we see is that across the tomato germplasm that we genotyped, we see at least 6 distinct subpopulations. So the land races classify in a group, the vintage classify in a group, the fresh market and the processing are distinct, and even within the processing, there appear to be distinct substructures, right? And these correlate with regional breeding programs. Okay, so California material represents a distinct subpopulation from Midwestern material, right? That may reflect adaptation, so selection for specific environments, or it may simply be residual from founder parents, right? We don't know that yet. I do want to point out that this analysis has already been done in a preliminary way for potato, and so if you look at the paper that was published out of Robin and Dave's work in BMC Genomics, and the link is here, you'll see that they've run this analysis and that you do see at least the chipping types in their own subpopulation relative to other potato types. Okay. Now, there's a reason, I'll come back to structure, but there's a reason that knowing the structure of your subpopulations is important when it comes down to association analysis and genome-wide selection, and I'll follow up on that in just a little bit. Another thing that you can get out of this kind of genome-wide survey analysis is an estimate of how linkage disequilibrium decays, okay? And this is important because it really tells you what the important marker density is for your crop. So in outbreeding species like maize, LD can decay within 1 kb. Okay? In inbreeding species like tomato, that's not the case. And in clonally propagated material like potato, it's also unlikely to be the case, right? You make a cross, you have 1 opportunity for recombination, but you've got whole parts of the genome that just don't recombine because your population isn't large enough. Okay? And so the bottom line is that in tomato, LD decays over like a 5 centimorgan window, right? So we don't need high marker density to capture a lot of information. All right? So that's what's demonstrated on the top of the slide for both processing and fresh market material. Now, another important thing that we see within processing tomato, and this is really reflected in the subpopulations we see with the structure analysis, is that we have significant gametic phase disequilibrium. Now, that's a big phrase right there, but really what it means is that there are genes on different chromosomes that appear to be linked because of selection. Okay? And really, that's what we do as plant breeders, right? We are selecting for combinations of genes across the genome. Okay? We can see this in a very striking way with tomato. Okay? So there are whole sections of the genome that are essentially co-inherited, right? Now, that actually has a very negative implication for our ability to use association mapping, right? And I'll explain why in just a little bit. Okay. First, I want to talk a little bit about some strategies in terms of genotyping breeding populations. So one of the things that was implied in some of what Dave was saying in his talk, and is also very clearly seen in the tomato material, is that we may not need to use that high-density array for all applications. In fact, we probably don't. So what I'm showing on this slide is a table on a chromosome-by-chromosome basis on the number of polymorphic SNPs in selected tomato populations. So this particular set here is for processing tomatoes, not fresh market. And what you can see is that if we look at chromosome 1, there's only really 75 polymorphic markers on that chromosome. But if we then impose some kind of a filter and say, look, I don't want to see markers that are closer than half a megabase, that number drops down to 20 because some of the markers are clustered. And if instead of using physical distance, we were to use recombination in a mapping population, what we would see is that a lot of the markers that are closer than 1 megabase simply never recombine because the populations aren't large enough. Okay? So if the markers don't recombine in our populations, they're not giving us new information, but we're still paying for that data point. Okay? So the point that I want to leave you with on this slide is that we can take these 7,600 SNP arrays and we can reduce it to a set of markers that are informative, highly informative to our populations. And then the next step is to decide how you're going to use it. Are you going to use it to facilitate some sort of a backcross program? So if you want to eliminate wild species genetic background in an integration program, you could maybe get away with just 48 SNPs that cover the genome, right? If it's really for genome-wide selection, you maybe want to go up to 384 or higher, depending on the population size, right? But the point is that we can create subgroups of SNPs and much more cost-effectively do what we need to do. Okay, so the Infinium array is an excellent survey tool to look at program diversity and substructure. If I were to make recommendations on how to use that survey tool, I would say make sure that all the germplasm in your particular program at the point where you start to collect phenotypic data gets genotyped with that tool. You can use that for possibly for association analysis. We'll put some caveats on that. You can use that survey to develop smaller, more cost-effective subsets of markers for subsequent selection and background genome elimination. And you can use that smaller set for very effective mapping in early generation material. Okay, with the number of markers really just dependent on the population size and your selection goals. Okay, and the other thing I want to point out is that in SolCap we've relied heavily on Illumina, but there are other companies. There's K Biosciences, for example, is a company where you can order a large number of primers. They can store those primers for you. You can then select subsets of them to run on material, and they basically are going to do it on a per sample basis. TACMAN reagents can be purchased in this way as well. And so a lot of this would be worked out based on maybe a core facility that you have access to or a service provider that you elect to use on the project. Okay. So take a breath here. What I want to do now is switch gears into strategies for integrating genotyping and breeding. One strategy would be to take a very, very large population with low marker density and genotype that. You would then select for informative individuals, and by informative individuals I mean individuals that maximize recombination in that population, and then you would phenotype that reduced population. Okay, so what you're doing is that you're go for a couple of thousand individuals. Close to 1,500 would be one package set. And simply, if there's minimal recombination in an individual, throw it out and only keep the individuals that maximize recombination. There's programs that can be used to do this. Okay. In fact, former student of Steve Tanksey's, who's I believe at North Carolina State University, has a program that can be used to facilitate that. So that's one approach. Another approach is the exact opposite. That is to phenotype a very large population, select the extremes, and then genotype those. Now, this is an approach that I think can work very well in some breeding programs, but the key here is that most of us, when we're breeding, don't keep both extremes. All right? But if you're talking about creating a training data set, it's nice to have the best and the worst. Okay? And so you may have to think about that idea just a little bit, and maybe for genotyping purposes, you are going to keep some of the worst. Okay? Hopefully that gets you thinking just a little bit. Okay, so let's move on to now integrating just a little bit more. So I've mentioned association mapping a couple of times, and I've mentioned the concept of genome-wide selection. These are similar approaches, but there's some statistical differences in terms of how the markers are treated, and I'm not going to go into detail on that. What I want you to understand is that populations that are consistent with breeding program designs are very amenable for this kind of analysis. But again, both population size and selection strategy become very important to success. Okay, so what I'm showing in this slide is like a simplified SAS code for association mapping. And so it's using the mixed model analysis of variance procedure. And the model is a trait. Okay, in this case, T1 is the trait. And that trait could be a least squared means, or probably better off to use a BLUP. And I'll talk about BLUPs in just a little bit. You then have a matrix that comes from the program structure. And so the matrix I've put in here is the Q matrix from the tomato population. So there's actually 6 subpopulations, and each row represents the probability that an individual line belongs in each of those subpopulations. Now, the matrix here is for 6, but you can't put them all in or you over-parameterize the model. So you always leave one out, but that one can be predicted based on the other 5. Okay? So the information is really there. And then there's a term for marker, and in this case, marker is considered a fixed effect. Okay? And then genotype and possibly replicate are considered random effects. And then we just cycle through all the markers. All right? So what I want you to understand is that the model here is making use of information from structure. Now, you can also bring in information from your pedigree, your knowledge of pedigrees, right? Arguably, we get more detail with the marker data, all right? And then, again, the power of this is going to be based on population size, recombination distance from the marker to the QTL, and the proportion of variance explained by that QTL, all right? Okay, now, strengths and weaknesses of the approach are kind of outlined in this slide. I've chosen 2 traits to focus on here. One of them is a fruit shape attribute. It comes down to the shape index, which is a height to width ratio. There's a gene called ovate, which was cloned in Steve Tanks's lab many years ago, which explains a lot of the elongation in tomatoes. Now, it turns out that ovate is essentially segregating across market classes. There are both alleles of ovate in fresh market material and in processing material. Even within regional breeding programs, it's still segregating. And so when we correct for structure, and that's what's shown here are just 2 of the structure classes, Q1 and Q2, you can see that that ovate allele is still segregating across the subpopulations. Okay, let me go back here, and I think, sorry, yeah, sorry about that. Okay, so ovate is an example of a trait that will work really well in terms of finding an association between marker and trait. Okay, it's segregating across all the subpopulation structures, and it's also a gene that explains a lot of the variation for that particular trait. Now, there's another gene which is called MAC-NOR, which is an interesting gene because it's been selected for and used in agriculture at 3 different times in history. So there are 3 alleles that have been used in agriculture. One of them, the alkabasa allele, has only been used in fresh market material. And so if we correct for substructure, we essentially lose our ability to find the effect on shelf life of the alkabasa allele because it's not segregating in the Q1 group, nor is it segregating in the Q2 group. Okay? And so once we correct for substructure, we can no longer find that gene. All right? In this case, Q1 turns out to be all material from Spain, from a specific region of Spain. Now, in contrast, if we look at the long keeper allele, okay, that's a gene and an allele that have been used in processing tomato breeding program, and it segregates within the Q2 group. So we can find that gene with a marker trait analysis, but the effective population size is reduced because the whole Q1 group is adding no information. Right? So I'm trying to present this in a way that's sort of intuitive. So the point is this, is that for association mapping and then therefore subsequent marker-assisted selection, the success is going to be based on population structure, segregation of the trait within subpopulations, allelic diversity of the trait, size of the population, and size of the subpopulations with that trait. Okay? And then finally the magnitude of the QTL, the proportion of variance explained by that. Okay? And so one of the recommendations that I have, and this may be a little bit provocative, but I would suggest that if you're planning to use this kind of analysis, that you maybe forget about what we would call standard association mapping with unstructured population and think about more of the nested association mapping type populations. Now, in maize and in tomato, these are all done using recombinant inbred lines, and I realize that that's not something you're going to be doing. But you could think about applying this to chain crosses within your own program. Okay? So there are ways that this could be used in the context of potato breeding. And then finally, I would emphasize again that large populations become really important. Okay. Now I'm going to switch gears to another example here, and this is an example dealing with bacterial spot resistance. And one of the reasons I like to present this example is because the crosses that we used are not the typical academic crosses where we take the best parent we have and the worst parent we have and we cross them. This is a population design where it's all best by best crosses. Each of the parents in here was bringing something to the table in terms of resistance to bacterial spot. In a couple of cases we knew about major genes or QTL, but in a couple of cases all we knew is that the lines were resistant in our trials, but we had no idea what the genetic basis of that was. So we created a cross, essentially a diallele, then we eliminated some of those if they were carrying too much fresh market germplasm, and then again intercrossed those. Okay, so it's not a traditional F2 type population, but we have a segregating population. All right, and we went to the field with a very large population, over 1,000 plants, and then we simply selected for the best and the worst, and then we carried those on. Okay, so in the case of tomato, carrying those on means we save seed, and then we're looking at the next generation. It's one more generation inbred. In the case of potato, you're just going to be bulking up that material and then doing an evaluation that's maybe based more on a plot basis as opposed to a single hill basis, right? So single plant selections in the first year and then replicated design in the second, and then the second year, because we're basing it on a plot, we can use a Horsfall-Barrett system to do our analysis. Okay, so now we've got a data set based on the extremes. We've got about 110 individuals that we've selected. We can genotype those, and then we can ask, okay, what models work? So if we use a single point analysis, we get a lot of noise. Single point analysis is not working. So we're seeing a lot of peaks that we don't think are real. Okay, so the position of one of the major genes that we know about is indicated as RX3 on this. If we take that single point analysis and we also correct it for population structure, we see that some of these peaks just go away. Okay, so what we were seeing there was markers that appeared to be tightly linked to the trait that were only really linked because of LD issues with the parent selection. Okay, if we then use haplotype analysis, so pairs of markers instead of single point analysis, we reduce the noise even further. And if we take that haplotype analysis and correct it for structure, we're really just seeing the one peak that we really know about on this chromosome. Okay, and so what I'm getting at here is that we can gain a lot of information by using a multi-point analysis or a haplotype analysis, and that we can reduce false positives by correcting for structure. Okay, all right. So, you know, in this particular set of best by best crosses, we estimated based on parent-offspring regression that the heritability was about 30%. Our realized gain wasn't that high. It was only about 13%. The population was biased towards the resistant end of the screen or the scale, but we kind of expected that because it was best by best crosses, right? Okay. And just to put it in perspective, you know, our average disease rating for those that were selected as resistant based on markers was 4, whereas the resistant control was 3.5. Okay, and again, the susceptible control was 9, which was much worse than the worst side of the population. Okay, and so, you know, in terms of how we can make use of these linkages in selection. It turns out that one of the major genes in the QTL are linked in coupling phase. And so one potential powerful use of the marker is to create, sorry, they were linked in coupling phase. One potential powerful use of the markers is to select for a coupling phase recombination event so that we can move those markers, those genes together. And that's something that can be done very effectively with markers. It's very difficult to do based on phenotypic selection. The other thing that we can do is we can reduce the selection cycle. In other words, we can be using the markers in the greenhouse in the off-season to select, make crosses, select, then go to the field. And so we can get 2 generations of selection a year. And this is where we can start to make up for that difference in the effectiveness of marker-based selection, 13% gain under selection versus the expected 30%, is by trying to get a shorter selection cycle. Okay. All right. Moving on to now the last part of what I want to communicate. And again, this is a strategy in terms of how we can make use of what we do well as breeders in the emerging data. I've hammered home the point quite a bit about population size being important. And after Walter's talk, there were several questions about analysis of the data and experimental design that lead me to think that the potato community is thinking about population or experimental design in a very positive way. So one of the things that we've done to maximize population size through experimental design is shifted some of our work to augmented designs. So for those of you that may not be familiar with an augmented design, I just want to give you a quick overview. Basically, the way an augmented design works is that we create our population is essentially unreplicated, right? But we replicate checks in such a way that we can account for different kinds of spatial variation in the field. And so in this particular design, we're accounting for variation. Let's see if I can get this mouse to go. We're accounting for variation in rows, in columns, and also in quadrants. So we have 4 rows, 4 columns, and 4 subquadrants, right? And so we can then, and then the X's are marking the spot for the replicated checks, but everything else is unreplicated. Now, the reason that this can work when we're doing analysis that's based on markers is because we're going to collapse it down anyway, right? It's the genotypic class that has replication, right? The actual line itself doesn't have to have replication. It's the genotypic class that we need to have replication on, okay? And so we use the overly replicated checks to account for field variation and make corrections as necessary. And so in this scheme here, if we had a randomized complete block design, we could handle 72 individual genotypes. If we use an augmented design, the way I've laid it out here, we can look at 128 genotypes. Okay, it's much more powerful from the perspective of looking at those marker trait linkages to have that level of genotypic replication. Okay, so larger populations but equal resources, and we can account for field variation. Okay, and so the data would look something like this, and I've already put in an example of a row and a column normalized, but you don't have to do that yet. Really, the first step is to ask, is there spatial variation in the field? And so what I'm showing you here is the results of an analysis of variance, and what you see is that, yeah, there's significant row within location variation. Okay, so we know that we have to correct for that. And so we can normalize our data for that row variation and then rerun the analysis. And what we see is that when we do that, that variation goes away because we've normalized our data to account for it. Okay? And so all of the significant effects are now removed except for the genotypic variance, which is what we're interested in as breeders. Okay? And so the idea is that we first run the analysis to ask, is there field variation and what accounts for it? Then we make the correction, and that it's that corrected data that we then would use for our integration of genotype and phenotype. Okay, so what I just showed you was an example that was based on considering the genotypes in our populations to be fixed. But I would argue that we might actually want to consider them to be random. They are random selections out of an infinitely large potential population. And if we are going to consider them to be random, then we need to be thinking about a different way of treating the data. And this schematic, by the way, is directly from a classic textbook on variance components, and the reference is there for you. Okay, and so if we consider the genotypes to be random, we need to estimate our traits based on a best linear unbiased predictor. And we can get estimated BLUPs from the proc-mix procedure of SAS very efficiently. There's two different ways of doing it that are shown here. The output of these two different ways is slightly different, but you can get what you want. And the upper method where we're just putting the solution, the slash solution, takes a lot less coding. The bottom method, we get the actual estimated BLUP, but takes a lot more coding to do. Okay, and so from that upper method, we get an estimate that's relative to the mean of the population, and then we can adjust. But the point I'm making is that we can take an augmented design trial, estimate the BLUPs, and then use that as our trait data for the marker trait analysis. Now, what's important to realize is that when you start to build up data sets over time that have overlapping sets of genotypes, is that you can estimate a BLUP for that whole data set, right? And so even though you don't have the same genotypes every year in the trial, you can begin to merge data, right? So the best linear unbiased predictor essentially allows you to predict the performance of a line that wasn't in the trial, in the same way that we can predict the milk yield of a bull, right? Okay. So this slide really sort of ties it up, and I don't want to go through all the details. What I really want to emphasize, so what I've done here is I've kind of color-coded some of the different aspects of a program. So the red represents analysis that gets done. The green represents really the activities of our breeding program in terms of making crosses, planting material, and collecting data. The blue represents the genotyping component, and then the yellow selection. And what I want to emphasize is that marker-assisted selection is going to be most powerful when we can use it to shorten the breeding cycle, and that's what I'm trying to show here. So if we can use it in this context to shorten the breeding cycle, over time we will make more progress. Okay? And so in conclusion, as breeders, we still need to recognize the importance of measuring phenotype. Point of analysis is going to be an important concept to grafts, so we're not necessarily going to be working with single SNPs, but pairs of SNPs, so haplotypes. Incorporating pedigree data or population structure into analysis improves the detection of trait associations and also the efficiency of marker-assisted selection. Our power goes up with population size. The number of markers we need really depends on recombination. Finally, phenotypic selection is effective. We know that, so we can't abandon the resources that we put into that. Mass can help if we use it to reduce the cycle.